All right. Oh, it's plugged in where you left it. You should text Hannah so Hannah has your number. Yeah, you turned it all the way off. Yeah, it's just a money suck. Attempted money suck. Well, I paid like a little bit of... Oh, forgot I'm live. So some of these pay apps, I give a little bit to them because I like the game, so I'm supporting their game. But the microtransactions ongoing, some people run up thousands of dollars. And, you know, They all do that. And
no, wait, but it's like a bootleg. It's not made by the Pokemon company.
oh, right, yeah.
Yeah, that's just dumb.
So
I also found this Pokemon Quest thing, and I didn't know it, and it just looked like a Pixelmon thing.
So we can work on recovering your password if you want a Pokemon Go back. Because you can, yeah.
It is a really mediocre game.
Okay. Because you can totally full on do it now properly, but that's okay. I respect your choice there. All right, I got 9 minutes to start. I'm going to start this lecture in 50 minutes. So my feed's on live right now. So I got to hurry and send the link to people. But
So wait, it's live so people are listening?
Possibly, yeah.
This conversation?
Yeah.
Cool.
In fact, did you get a notification that you might have in your email?
I did get an email that said on my way home.
Oh, I sent you that message, yes.
I know.
I misspelled it because I was at a stoplight because I wasn't texting and driving, but yeah. Yes. What? Oh, thank you. All right, let's see what we're looking at now. Oh. Here it looks like I have product in it. Oh, well. Oh, there's no viewers right now anyway, so that's perfect. So let's go here. Oh, yikes. Sorry if that clicking is super loud. What? Right. Right. YouTube. 4 minutes. Okay. Those are set up. Be minimizing the click problems. See if I can check my levels right here. Oops, sorry. This chair is not the greatest. And this is me with my own sound muted, I think. Yeah. Oh wait, here's what I want to do. I want to take an audio source. I want system sound. Okay. Am I going to know? I have no audio preview set up. I have system sound turned on. Game source, right. Copy link to clipboard when starting a broadcast. What? I didn't have to go out and find it. Yay. Okay, that's fun. So audio. Oh, wait, okay. I guess I'll leave that on default speakers. See, I don't want... Okay. Probably have to investigate more of this. As I learn to use it more, as you can see, I abandoned the green screen. Because that wasn't working right now anyway. Do I need more light on me? Okay. Okay, in theater, if you have to ask, the answer is always yes. Pretty much. Unless you're supposed to be in the dark. I don't have really great... Oh, God, no. There we go. All right, I've got 1029. I'll probably be using this some on here, too. Wait. Okay, good. I can see the audio level, so that should be fine. Okay, it's 9:30 Central Standard Time, so let's get going. Welcome to October 8th. This lecture here, we have mechanisms of microbial genetics as the next chapter we'll be going into. I might come back for the actual chapter 8 later on, but probably not. If there's anything in there I feel like I need to tell you, I will come back and collect that and give that to you, but I probably won't add an assignment to it, have an assignment associated with that. Or I'll fold it into another assignment, whatever. It's the fermentation that would be of particular use, as well as some of the geochemical cycling, because then I get to talk more about nitrogen fixation, which is fun to talk about. Oh, and okay, so let's get to this. Let's come on down into here. So the essential element that connects all of life that we know about is DNA. And such as the elephant and the Escherichia coli, they have a lot in common when it comes to microbial, when it comes to molecular genetics and how that works. There was a particular French scientist, Monod, that said that was famous for quoting that, for saying that. Anyway, so in this chapter, we're going over the functions of genetic material, DNA replication, RNA transcription, protein synthesis. So the central dogma of molecular biology in the middle here, and then some mutations, how genetic diversity is achieved in prokaryotes, which I've already kind of spoiled because we've talked about horizontal gene transfer already, and of course operon theory when it comes to gene regulation. So that'll be in class, but yeah, that'll be in lecture. I mean, we'll be physically present in the classroom when I go over operon theory, because this whole gene regulation thing is going to be a question they're definitely going to ask you in the future. And genetics is a possibility, and but there's just some basic, like the lac operon and the trip operon are the standard gene regulation models that you need to be able to discuss, and we will go over them probably Friday, unless we can get to it Wednesday. We'll see. I don't want to go driving through this too fast, even though largely a lot of this you're going to get from you got from 115 or are getting, and cell biology, genetics. We do go over the same ground quite a bit, so I don't want to belabor that too much. But anyway, so section 1, the functions of genetic material. So explain 2 functions of the genome, explain the meaning of the central dogma of molecular biology, and differentiate between genotype and phenotype, and explain how, we'll get 2 viewers, yay, oh, welcome you too. Differentiate between genotype and phenotype, and explain how environmental factors influence phenotype. Okay, so this is the central dogma of molecular biology. DNA is transcribed into RNA, which is translated into protein. This is the flow of information from our genome to actual products. DNA encodes messenger RNA, which in turn encodes protein. We do already know there's a way to go from RNA to DNA in the form of retroviruses, and that there is a lateral transformation that can happen between proteins when we talk about prions. But largely, DNA to RNA to protein. That is the flow of information. Clinical focus, part 1. So we have Mark, a 60-year-old software engineer who suffers from type 2 diabetes, which he monitors and keeps under control largely through diet and exercise. So he scrapes himself, his lower leg, walking through blackberry brambles. That means he was like doing gardening in shorts or something, doesn't it? He continued working all day in the yard and did not bother to clean the wound and treat it with antibiotic ointment until later that evening. For the next 2 days, his leg became increasingly red, swollen, and warm to the touch. It was sore not only on the surface, but deep in the muscle. After 24 hours, Mark developed a fever and stiffness in the affected leg. Feeling increasingly weak, he called a neighbor who drove him to the emergency department. Did Mark wait too long to seek medical attention? At what point do his signs and symptoms warrant seeking medical attention? So when it came to having to go to the ER, this is all my opinion, obviously. I mean, at least he went then at that point. And I don't know, my own, if it was on my kids and there was they had something that was becoming red and swollen from a scratch, we would have gone to at least to the pediatrician or something long before that. That's 3 days later total. Anyway, but it's hard to, so there's an aspect of, I'll just say the words, toxic masculinity that drives especially us in America to refrain from seeking medical treatment until the last possible minute. But part of that is a function of our lack of, like really good health care. He's a 60-year-old software engineer. Maybe he has health care, maybe he doesn't. But, I'm guessing in other countries they don't have this issue. They can't because they have much better health care outcomes than we have. So they can't go too long, right? Yeah, because they have better outcomes. Okay. What types of infections or other conditions might be responsible for marked symptoms? I guess we'll find out, right? Obviously, it's going to be some kind of microorganism. And I swear my hair does not have product in it. That's just water. I was hoping to be a little more dry by now, but apparently not. All right, anyway. So genotype versus phenotype in the context of intro to biology, you've probably gone over this before. And it's not really different in microbiology or with microbes, except by the fact that you can have very different phenotypes from your organisms based on their environmental conditions. So the genotype is going to be the actual genetic composition of the organism, so the type of alleles that they have. And then the phenotype is how those alleles get expressed, how those genes get expressed. And in bacteria and microorganisms, you can have dramatically different phenotypes based on different conditions. The go-to example for this, the really quick one, is Serratia marcescens, which is the one that will grow red, express a red pigment when it's grown at room temperature. But when it is grown at 37 degrees, like in an incubator, or at body temperature, it does not express the red pigment. So completely different appearance, the same genotype. And there's a lot of these. There's one of our routine organisms, and the name is escaping me right now. It might be one of the Pseudomonases, but there's one of them that will spontaneously or randomly between generations change its morphological appearance as a colony, and it makes it look like your organism is contaminated when it's really not. And I've been fooled by that more than once. So I kept thinking I had a strain. Oh, I need to remind myself, make sure we're getting our freeze cultures frozen. Okay. All right, so again, genotype to phenotype, there are differences in gene expressions results in different phenotypes. And when we're talking about diploid, large organisms, you're talking about that are dominant, recessive, the whole Mendelevian thing there too, resulting in different phenotypes. Like 6 genes for eye color, I think it is, or is that skin color? Yeah. So, Sigma Xi, their little eye on ethics segment in this chapter, is talking about the use and abuse of genome data. So, you can go ahead and voluntarily get your whole genome sequencing. Whole genome sequencing is not a routine thing yet. That's still over $1,000, or maybe as cheap as $1,000 now. So having an insurance company pay for genome sequencing might not be particularly useful. But anyway, this Eye on Ethics is about the use and the abuse of genome data. Right now you can get, so if you do Ancestry, 23andMe or whatever, my wife and I did 23andMe, you get ancestry data and you get some medical data, but these are all based on single nuclear polymorphism, SNPs. And these SNPs are just tiny portions of the overall sequence, generally in an area that's non-coding, that's in that region, but not exclusively. But anyway, what should people be able to do with that data? Well, we know one thing, what people are doing is we're solving some crimes. We had the Golden Gate Strangler case was solved because of relatives of that individual had uploaded their genetic data or had provided samples that were analyzed. So, but anyway, should be required by law. This kind of health sequencing. We're rapidly coming back to where we're going to get rid of pre-existing of the prohibition against excluding people for pre-existing conditions. That's bound to happen within this next year. Maybe depending on what happens next month, we'll see. Make sure you vote. We got to, participate in this democracy if we're going to have a country left. But anyway, okay, hopefully that won't be an issue. Just got a message about high CPU usage. So, sorry. We, should you be able to be charged or rejected based on your genetic data? And do companies have the right to compel that data? I'm pretty sure they're going to get that ability. But right now it's prohibited, but it doesn't cover life, disability, or long-term care insurance policies. So there's another story that was recently, you probably saw it in a feed somewhere. Here we go. So there's companies that are wanting you to hand over your Fitbit data. And, well, okay, this is Washington Post, so it's going to say, so it can make more money. And yeah, that's ultimately to reduce their costs, right? So this life insurance company, so this is an option. They're not making it mandatory yet, but using fitness trackers. And I'm going to tell you, I already voluntarily do that. My session has expired with Humana. I already voluntarily do that. Oh, you're going to see that I haven't worked out much lately, but that's okay. We had our break, right? So we have all these points and you can buy stuff with it or whatever. So it's really probably not that hard to get a lot of people to just voluntarily give up this data anyway. There's my weekly view. Yeah, it's not that great. And so some of these tools in here, in my opinion, can actually be helpful in maintaining a health goal or whatnot, but I don't think it should be compelled. But I'm okay with the idea that companies are using them to provide discounts or offer discounts to people. Let me peek in on this right quick. Oh, wait, I would have a notification if that was true. Sorry, I know that typing gets really loud as well. All right, moving on. All right, this was my reminder to show you that other page. Health insurance companies and you. I don't, just to be also, hopefully be clear, I would never want to see people, companies be able to kick people off for pre-existing conditions. But again, we're going to experience those days again, I expect pretty soon. All right, section 2, DNA replication. So the first leg of our Central Dogma. So in this, the learning objectives here include the meaning of semi-conservative DNA replication. Again, for if you ever take a GRE specialty, I got to talk about those at some point, or the MCAT or whatever. These are questions that are going to keep coming up. Probably even for, this is something that can easily be asked if you're in a molecular graduate program of some kind, and you practice for your oral boards or doing your oral boards. Explaining the meaning of semi-conservative DNA replication is definitely an easy question to ask that can trip some people up if you don't keep up on your fundamentals. And that's, I don't know if I told you in class yet, but graduate school, at least on the molecular side of life that I experienced, is so much easier if you remember your fundamentals and you get your fundamentals down really well, because the majority of what you wind up taking for coursework are going to be just higher levels of detail of the same things you already learned. You're building on that same scaffolding and just enhancing the depth of your own understanding and the breadth of your own knowledge. In chemistry, they use the analogy of a train, and that from your first 201 or whatever chemistry course, you got to keep up and stay on the train in order to be able to make it through successive courses. It's similar in molecular biology, but I think of it more as a tree, that you start with just a skeleton of a tree or whatever, and you just hang more stuff on it as you go. I don't know. The train's probably a better analogy. Anyway, explain why DNA replication is bidirectional and includes both a leading and a lagging strand. Explain why Okazaki fragments are formed. Describe the process of DNA replication and the functions of the enzymes involved. Identify the differences between DNA replication in bacteria and eukaryotes. Explain the process of rolling circle replication. So, in 1953, we determined the structure, the anti-parallel nature, as well of DNA. So the following question was,
what and how is this molecule replicated or duplicated. And these were the models that were suggested. So the first idea is that it's conserved. And so conservative meaning the original DNA molecule stays intact at the end of the process, and you make a complete new copy of them, and that makes their own double helix over here, while your parent strand stays over here. So that seems like a fairly intuitive conclusion, right? Another model was the semi-conservative model, that since you've already separated these two parent strands and you're synthesizing new strands, well, why don't they just stay together? And like that. So you have your original single strands, the original parental strands are split up with new strands all along. Which, so, just something I thought of once, and there's probably not much of a point in going on long about it, but that would mean that ultimately there are pieces of ancestral DNA that go back thousands of years, I would think. There's obviously no intact chromosomes, but little fragments and snippets of it, of the same nucleotides, some of them bound in the same way, that go back to ultimately to the first organisms, right? The first DNA containing things. But it would look more, anyway. So another idea was that there's no rhyme or reason necessarily to the parental or the daughter strands, and it's just randomly distributed. Okay. Well, here's the experiment that solved it. Again, this is another experiment that will come up for you in the future in different ways. So this is with E. coli, using E. coli DNA, using a stable isotope of nitrogen, nitrogen-15, and along with native nitrogen. So first, they grew at least one or two generations or so of E. coli in a medium where their nitrogen supply was exclusively nitrogen-15. So meaning that when you put it on the ultracentrifuge in this, I think this is a cesium chloride gradient. Of course, I put my microphone right in front of my view, but I have all the words here too. When you spin it all that DNA out, you get a single band, and it is only because of all the nitrogen in the DNA is of the heavy isotope. So for any of these potential models, this is what you would expect, I guess, because all the DNA is the same. So then you grow at least one generation's worth. So this is, you're looking at your inoculum started with your heavy nitrogen strain, and you let it go like one generation. I'm sure they had to do a number of attempts on this to, make the protocol work out. And then you grow it with regular nitrogen-14, the most abundant isotope. And then you extract the DNA and you spin it down. So you should have what you would expect here is, if it was conservative, where the parent strands stayed together and the daughter strands were separate, is that you'd have two separate bands. One of DNA composed of nitrogen-14 and one with the DNA that has the nitrogen-15. This is not what was observed. So right at this experiment, you ruled out this conservative model. What actually happened instead is that the strand, the DNA got separated out. It was a little bit lighter than in the previous run with the nitrogen-15, but it's still a single band. All right, so then after that, you grow it another generation with nitrogen-14 only, supplementation there. And then you spin it out again. And what they observed is that they had the same, we'll call it the purple band here, for the intermediate weight, but they also had this other band that was the lighter variety of DNA, using only nitrogen-14. And what you would expect in the dispersive model, if it was still just randomly different segments of DNA, that it would still all be together as a single band or a little smear or something down here. Instead, Meselson and Stahl supported the idea that it was semi-conserved using stable isotopes and how it sedimented differently in an ultracentrifuge. This is what I wanted to do with my DNA that I got from my Eustalago, where I only gave it, the only nitrogen source available to the Eustalago was from the nitrogen 15 in the air, dinitrogen 15. There was no nitrogen source in the media. You could argue there were trace contaminants or whatever, but it still couldn't have resulted in the growth I observed. So I wanted to do the same thing. I wanted to spin the DNA down compared to, first nitrogen rich media, normal growth. And I wanted to compare that to grown with nitrogen 15, dinitrogen 15 only available in its little tube. And then nitrogen 14, regular atmospheric composition air in an isolated tube with no other source of nitrogen. I wanted to do those comparisons because we had an ultracentrifuge down there, but it didn't work out. So I had to do something different that was took a lot more source, a lot more culture. Anyway, Meselson and Stahl. Okay, so DNA replication in bacteria. This is faster. It's about 1,000 nucleotides per second. And it is bidirectional from the origin. There's a replication fork that I will mention. There are multiple DNA polymerase molecules, and these are kind of what they are that we're talking about at this level of detail that we're at right now. So we have DNA polymerase 1. It primarily functions in repair. DNA polymerase 2 is also primarily in repair, but in a different area. DNA polymerase 3 is required for DNA synthesis. Okay, so, and the story on these, the why it's repair and a synthesis is that DNA polymerase 1 was the first one isolated. The same issue with the photosystems when you talk about photosynthesis. And it was first thought that that was the primary replicative molecule, but it turned out not to be. Okay. Now, dNTPs, you know what? This whole time I could have had this open for me. Oh, no, not that. Dagnabbit, sorry. Oh, this one. That's what I want. Okay. In case I had any notes I wanted to mention. Okay. So the fuel for replication, for DNA replication, and indeed for most things that we have going on, is our dNTPs. So it's not just ATP, it's all of the nucleotides and with 3 phosphates. It's dideoxy nucleotide, triphosphate. So this gamma phosphate, this bond right here, is the one that's fueling replication. That's important to remember because you always need to remember your dNTPs. Okay, let's see. Here we go. The origin of replication, the ori in Escherichia coli. Not that anyone ever watched Stargate much, probably, but that's an old reference now anyway, the ori. So the origin in prokaryotes, you have about 245 base pairs. It's rich in AT pairs. We have, I'm talking about initiation of DNA replication. So you have the ori. That's where things are binding. Then we have topoisomerases that are able to manipulate the packaging, the supercoiling of the chromosome. And specifically, topoisomerase 2 is DNA gyrase. That is the enzyme that relaxes supercoiling. In fact, it also needs to wind it the other direction. You need negative supercoiling in order to initiate DNA replication. And this process is not free. It requires ATP. Then we have our helicases. These ones separate DNA strands. This also requires ATP. And so here's a couple of antibiotics that inhibit this process. It inhibits helicase specifically. And resistance to those antibiotics involves mutations to the topoisomerases. So Cipro is probably one that people recognize. more readily than novobiocin. And we might be doing that lab this week, so Wednesday, looking at the antibiotic discs. All right. So this all gets arranged in this replication fork. And we have, here's our helicase doing the gyrase doing the negative supercoiling. You have the helicase doing the unzipping. We have oh my God, the sliding clamp is so cool. And I'm going to show you the video on that. But anyway, and I didn't even know it existed until molecular biology. In the intro to microbiology, I had no idea. But there's this clamp that slides on to the DNA polymerase that makes it a lot faster, increases its processivity is the term for that. So anyway, remember you can only add nucleotides in the 3' direction. So when you have a replication fork, there's one direction that is going to have to be discontinuous, and that's called the lagging strand. But the other side can be continuous, and that's the leading strand. So the leading strand on one side of the fork is the lagging strand on the other side of the fork. So you have one side that can go, right, so continuous synthesis on the leading strand, discontinuous synthesis on the lagging strand. That's the important difference there. So the little fragments here that have to be filled in from a primer, those are Okazaki fragments. They get DNA polymerase 1 is the enzyme that will come in and fix these little gaps here where the RNA primer was. So we need an RNA primer, and we use an RNA primase for that. And then that allows the double-stranded RNA-DNA complex is what allows the polymerase 2 to bind, and then it will go a short distance until it butts up against the previous fragment, which is getting repaired by the DNA pol 1, and then DNA ligase will finally fix the backbone here, essentially. Okay. There's a video I'm going to show that has all this in pretty pictures. So that's at the initiation. Well, this is more than just initiation, right? So elongation. I want to mention, too, that these two polymerases are connected by a protein complex you can't see here. It's called the gamma linker. And it's also the one that has the clamp loader. And it literally clamps, loads a, like a ring protein on and clamps to the DNA pol 3 and the double-stranded section that is being elongated and the template. All right, so elongation is that process. Let's see, anything else? Holds pol 3 in place, yes. Processivity, yes. Topoisomerases prevent overwinding by nicking and resealing. What's also not pictured is a bunch of what's called single-stranded binding protein. And when this replication fork is opened by the helicase, those single-stranded bits of DNA are now vulnerable to other enzymes that will chew it up, that will chop it up. This is much more of a problem for prokaryotes because they don't have a nice membrane-sealed nucleus to do all this in. All right, so the DNA pol 1 also, I did not actually say it, but it removes this RNA primer through an exonuclease activity. And then the DNA ligase catalyzes the phosphodiester linkage back here to repair this. Okay. So termination, there are a couple of different ways to terminate replication. Oh God, I'm going to have to cough. Crap, sorry. Oof. I shouldn't still be sick. I was a little bit sickly last week. And, this is not from your book. I'm not going to force you to know this, but there's a process for replication termination and separation of these two strands from each other. Because so I guess this is a preview of when you take more molecular courses. So it'll be interwoven with itself when it makes this copy. They'll be linked together like a little chain. And there is a process to excise. So basically, make a double-stranded break in one to let the loop of the other one out, and then it's repaired. Okay, so here's a summary of the mechanisms of DNA. Okay, it's 1102 or 1002 for where you are mostly. So anyway, the molecular machinery involved in bacterial DNA replication. We have DNA polymerase 1 has exonuclease activity, removes RNA primer, replaces it with newly synthesized DNA. So that's important to know. DNA pol 2, that's the primary replicative enzyme, and adds nucleotides onto in the 5' to 3' direction. So it adds the new one at the 3' end. Helicase is the enzyme that opens the helix by breaking hydrogen bonds between the nitrogenous bases. We have ligase that seals the gap between the Okazaki fragments on the lagging strand to create one continuous DNA strand. We have primases, which are the enzymes that synthesize the RNA primers. Single-stranded binding protein binds a single-stranded DNA to prevent hydrogen bonding between DNA strands, reforming double-stranded DNA. Sorry, that's the other reason. So you don't want it binding weirdly too. So you're protecting it and you're isolating it from forming a spurious new hydrogen bonding interaction, making a weird hairpin or whatever. Sliding clamp. This helps hold DNA polymerase 3 when nucleotides, DNA polymerase 3, in place when nucleotides are being added. DNA gyrase relaxes supercoiled chromosomes. It helps relieve the stress on DNA when unwinding caused by breaks and then resealing the DNA. And then topoisomerases type 4 induces single-stranded breaks into the concatenated chromosomes to release them from each other and then reseals the DNA. That's different than how plasmids replicate, which we'll go over later on. All right, let's see if this will work. Oops. Can I play it from in here? Try. I don't hear anything. Do you? Do you hear anything? Yikes. Oh, well, that's not what I wanted anyway. All right. Hey, 4 viewers. Yay. Okay, so let's go to here. Oops, sorry. This is from the DNA Learning Center. Let's see if I have any sound. Crap. Might have to ditch this. Oh, wait. Oh, that's why. Hey. Oh. That's so cool. So that's the gamma linker right here. These are the two polymerase 3s, and that's the clamp on the back of it. And then we have the topoisomerases and the helicases. Well, only one of those are pictured here, and this is the clamp loader right here. Here we go. Oh, there's a Uploader is off. Look at that clamp. All right, that is here. Okay, we can move on from there. Whoa. Sorry. All right, so in eukaryotes, not only are the polymerases named by Greek letters instead of numbers, which is really freaking annoying. I don't know. I guess you can help keep them separated or whatever. But we have, let me go back to this screen here. There we go. We have multiple origins of replications in eukaryotes. But part of the trade-off here is that the, 30 to 50,000 origins. And but the processivity is only about 100 nucleotides per second instead of 1,000 in prokaryotes. So eukaryotic chromosomes are typically linear and each contain multiple origins of replication. I guess that's the main takeaway here, though there are some prokaryotes that have multiple chromosomes and that are also linear. So DNA polymerase delta, wait, that's what that is, yeah, delta. And then epsilon, now I'm not going to go over that. That's eukaryotes. They have different polymerases. The process is similar though. So that video stands. You have to learn all about your eukaryotic polymerases elsewhere. I don't know. Anyway, let's see next. Oops. So these linear chromosomes, when the DNA polymerase reaches the end, it falls off because it can't complete the last little bit. So we have in eukaryotes repeated segments called telomeres, and the enzyme telomerase is able to come back and helps extend that and synthesize replacements for that. And so here, this is in a lagging strand direction. So you have a primase and DNA polymerase that comes in and fills this in. So when you're born, when you're fertilized, when you're an egg and you get fertilized, you'll have telomerase active. You'll have it active for a long period of time after that in development. But then at some point it gets switched off. And when this was discovered by Elizabeth Blackburn, I don't know if she thought this, but some people certainly did. They thought that the telomeres, so you have up to 100 or 1,000 of these at the end of each of your chromosomes, and that each time the cell replicates, it loses one or more of these tags at the end. And so that's kind of like a fuse to senescence or cellular death, barring any other kind of injury to the cell. But this telomerase, they thought that was all you need to do is activate telomerase to be, immortal, effectively, cellular immortality. But that's not the only thing going on. Senescence is not. Cells can go into senescence for reasons other than running out of telomeres. Oxidative stress being really important to that. So these are active in germ cells, and it's also still active in adult stem cells. So that's enough about telomeres. So the basic comparison of bacterial and eukaryotic replication include singular single circular chromosomes, and that's true for tons of prokaryotes. Eukaryotes have multiple linear chromosomes. We have one ori per bacterial chromosome. Eukaryotes have many, many more. The processivity is 1,000 nucleotides per second in bacteria, 100 nucleotides per second in eukaryotes. Telomerase is not present in bacteria. It is present in eukaryotes now. So you're like, well, hey, how do these prokaryotes with the linear chromosomes, how do they handle the ends? I don't know. RNA primers are removed in bacteria by DNA polymerase 1. It's RNase H in eukaryotes. DNA strands are elongated by DNA polymerase 3 in bacteria. Make sure you know that part. And then these polymerases in eukaryotes. All right. So I think finally in this section 2, we're looking at rolling circle replication. Oh, excuse me. This is how plasmids replicate. And so there's a nick made at the double-stranded origin. And polymerase 3 is involved. So this just starts rolling off. And gets replicated here. And that's all there is to it. So the process of rolling circle replication results in the synthesis of a single copy of a single new copy of the circular DNA molecule as shown here. Like I said, plasmids engage this. So there doesn't need to be a separate step to release the concatenated strands. And yeah, so the single-stranded origin and a double-stranded origin. And then, yeah. All right, RNA transcription. And we got 6 minutes. How much do I want to get into this? RNA synthesized using DNA as a template, transcription between prokaryotes and eukaryotes. That's all. I'm going to stop here, I think. I'm going to do section 3. I guess I'm hungry. All right. Anyway, we'll see y'all on Wednesday. All right. Oops. Yes, I do want to save changes. Here we go. Oh, there we are. Well, thanks for viewing. I'll see you on Wednesday.